Greetings, parasite ecologists. You know, there's a funny thing about being a parasite ecologist. Other ecologists go out into the field, by which I mean outdoors. They might sit on a rock in the forest and watch as things happen. Two big male deer compete to kick one another out of that forest stand. Or maybe that ecologist will be scuba diving on a coral reef and they'll watch a damselfish zip up to another fish and bop it, trying to get the other fish away from its algal garden. You might think that being a parasite ecologist is different. I can't sit on the forest floor or hover mid-water in a coral reef and say much about what's happening with the parasites all around me. But when I open up a host and I peer inside, all the same stuff that you might observe between two deer or between two fish, all that stuff is also happening among the parasites. Our forest, our coral reef, the habitat of parasites is inside the host. And once you find the parasites in there, you'll also find that they behave like deer or fish or any other free-living species. Let me show you what I mean. From previous videos, we know that adult and larval tapeworms behave differently within their host, and we know that they can get different things from the different locations that they infect. The anterior portion of the gut is a very different habitat than the posterior portion. But so far we've been assuming that each parasite can freely choose where it wants to infect. What happens when there's more than one worm in a host, and now they need to learn how to share? For this question, we're going to go back to our good old model system, Hymenolipus diminuta, the rat tapeworm. Let's begin with a couple of definitions. First, I want to define competition as an interaction between individuals brought about by some shared requirement for a resource that is in limited supply. Of course, if you don't have the pie to yourself, if you need to share it and divide it up into smaller slices, it's going to lead to a reduction in growth or reproduction of individuals competing for those slices of pie. Please also recall from your prior ecology training that a population is a group of organisms of one species, and a community is a group of organisms of more than one species that have trophic or spatial relationships with one another. They're eating one another, competing with one another, or living in the same place. Let's dive deeper into competition to define intraspecific competition as competition between individuals within one population, meaning individuals of a single species, and interspecific competition, which is competition between two different species in a community. Okay, before we see how intra and interspecific competition might work for parasites, let's talk about how to measure it. Tapeworms are really long, and they have only one attachment point. Usually, peristalsis, or the muscular contractions of the intestine, push poop ever posteriorly, a never-ending tide of poop that causes the long tail end of the worm to be pulled down toward the anus from its attachment point at the scolex. You might imagine that a tapeworm being attached at the junction of your small and large intestine and then being strung out along your whole colon. Now, this is a human, of course, but it works about the same in any vertebrate gut. What's the location of this worm? Small intestine, large intestine? What if peristalsis slows down and it can contract itself to occupy a shorter distance of the gut tract? We could measure the midpoint of the worm, but that would be difficult and it would change. However, there is one very simple measure. Where is your scolex attached? Some tapeworms are attached permanently, others aren't, but they can't move their attachment point very easily or very quickly. The scolex isn't necessarily where all the nutrient absorption action is taking place, but its position is easily comparable from one worm to the next and from one host to the next, and it's something that we can measure in a dead host. For example, a rat in one of our experiments. So to measure the tapeworm's position in the gut, we're going to measure the percent of the gut anterior to the place where the scolex is attached. So if the scolex were here, we'd say 5% of the gut is anterior to the point of attachment. Here, 30%. Here, 50%. The closer you are to the host's anus, the greater the proportion of gut there is between you and the beginning of the intestinal tract. Okay, so our definitions are in place. Now, what happens when we put more than one worm in a gut? Do they compete with one another? Are they scrabbling to split that pie? Or maybe because they're parasites, the pie just gets bigger. You could imagine that maybe if there are two of them, they just take more from the host. There's more than enough for both of them. Maybe parasites don't compete the way that deer in a forest or fish on a coral reef do. Let's see. The trick for seeing whether any organisms compete with one another is to measure what happens in the presence and the absence of a potential competitor. If everything's the same, whether the potential competitor is there or not, there's no competition. If things are different when the potential competitor is present, that indicates that competition is happening. So how do we do this for tapeworms? Well, we know that the gut habitat is really heterogeneous. Food and environmental conditions are very different in the anterior portions compared to the posterior portions. It's reasonable to think that some gut microhabitats are better than others. So maybe let's see whether worms pick different positions in the presence of competitors than in their absence. Let's get some rats, experimentally infect them with Hymenolipus diminuta. We'll either feed them 5 metacestodes inside of beetles that will give us 5 adult worms in the rat gut, or we'll feed them 10 metacestodes, or 20. Then we'll let those worms grow into adults, euthanize the rats, dissect the rats, and measure where in the gut tract each of their scolices is positioned. Remember, these are all single-species infections. All the worms are Hymenolipus diminuta, but the infections have different intensities, which we, as the experimenters, control. Along the x-axis is the percent of gut anterior to the attachment point, and on the y-axis is the percent of worms. In the top panel, you can see 5 worm infections, then in the middle, 10 worm infections, and on the bottom, 20 worm infections. In this light 5 worm infection, it appears that most worms are choosing to put their scolices at the 20% mark. But as we add worms to these experimental infections, their scolices are getting spread further and further down the intestinal tract. We add more competitors, and things are changing, indicating that competition is taking place. You can see that the optimal location for the scolex is moving rightward as we go from a low-density to a high-density infection, suggesting that as we accumulate more and more worms, some worms are being displaced into suboptimal habitats in the posterior portions of the gut. There is competition for the optimal location. When worms have their choice of locations, they all choose to be in the anterior portions of the intestine. As there are more and more of them, more worms are forced into the substandard posterior positions of the intestine. But on top of the influence of intraspecific competition on the position of these worms in the gut, we also see impacts of intraspecific competition on the parasite's fitness. This is sometimes called a crowding effect. Intraspecific competition becomes more intense as worm density increases, and there are more individuals competing for the same number of limiting resources. This forces the worms to both occupy suboptimal habitat and share available resources. Basically, everyone is splitting the same pie, but the more worms there are, the smaller the slice for each worm. To see whether crowding effects are happening, let's also measure the body size of the worms in the low, medium, and high density conditions. Here on the x-axis, we have the number of days post-infection. You expect worms to get bigger as the number of days post-infection increases and the worms mature. And then on the y-axis, we have the mean weight in milligrams of individual worms. Of course, the bigger a worm is, the more eggs it can produce, and the higher its fitness. You can see that in the lowest density treatments, we have much bigger worms than in the highest density treatments. And that's because not only are these worms being displaced into suboptimal habitat at higher densities, they're also trying to split the pie with a greater number of worms. The more worms there are in the gut, the less there is to go around for everyone, and the smaller those worms end up being. That's what we call a crowding effect. Okay, worms are using different positions in the gut in the presence of competitors, and they also attain smaller body sizes. Seems pretty clear that intraspecific competition is happening for Hymenolipus diminuta. Let's now talk about interspecific competition, meaning competition between 2 different species, not just among individuals of one species. Let's take the community ecology perspective and ask, what happens when there is more than one species of worm in the host? To do this, we're going to pair up our workhorse, Hymenolipus diminuta, the rat tapeworm, with Moniliformis dubius, which is an acanthocephalan parasite, a new kind of worm parasite. We'll talk about acanthocephalans a few videos down the line, but for now, just know that it is a different species than Hymenolipus diminuta. We'll set up the same kind of experiment that we did for intra-specific competition to find out whether Hymenolipus diminuta and Moniliformis dubius do inter-specific competition. First, let's get to know our potential competitor. Does it also like the anterior gut positions that Hymenolipus diminuta likes? Let's do the same kind of experiment with Moniliformis dubius. A small number of these acanths, a moderate number, and a large number. And yes, indeed, when they are at low densities, they seem to like roughly the same position in the gut tract as Hymenolipus diminuta does, probably because that's where the pH conditions are the best and the amount of nutrients available for absorption is optimal. The more acanths we add, the more worms get displaced into suboptimal habitats outside of that preferred range. Now, to test whether Hymenolipus diminuta and Moniliformis dubius are competing, let's compare what happens in a single species infection compared to a two-species infection. When we grow Hymenolipus diminuta in the presence of Moniliformis dubius, we see that the place where it tends to wind up is in the posterior portion of the intestine. Curious. Let's see if the same thing happens when we look at Moniliformis dubius. For Moniliformis dubius, we see that when it's grown alone, it also tends to attach in the anterior portion of the gut. And that's also the same thing that happens when it's grown in a double infection with Hymenolipus diminuta. So when we grow these two species together, Hymenolipus diminuta moves, but Moniliformis dubius doesn't. What we can infer from this is that Moniliformis dubius is the superior competitor. It's better at getting those anterior attachment sites that both species want, and it displaces the inferior competitor, our poor rat tapeworm, from its preferred habitat and throws it into habitat where it doesn't do as well. We see evidence for interspecific competition here. Let's look at all the interspecific competition data on the same set of axes. On the x-axis, just like before, we have the percent of gut anterior to the attachment point. On the y-axis, anything that's positive represents the number of Hymenolipus diminuta, and the columns that are negative represent the number of Moniliformis dubius. You can see that the histogram for Moniliformis dubius is the same in single and double infections. But for Hymenolipus diminuta, it's very different in those single and double infections. From this, we infer that Hymenolipus diminuta is being displaced from those preferred habitats at the anterior portion of the gut. So we see competition between these two species. The acanthocephalin is the superior competitor, and it displaces our poor little rat tapeworm. Our rat tapeworm is forced to occupy suboptimal habitat, and it has reduced fitness in that habitat as a result of competition with the acanthocephalin. Now, you might think it can't be that typical for two different parasite species to wind up in one gut, right? Wrong. There are tons and tons of intestinal parasites, not just tapeworms, but lots of other things that compete for space and resources inside of the intestine. This is the mean number of intestinal parasite species per host across a bunch of different fish, birds, and mammals. You can see that birds, and in particular water birds, are real hotbeds of gut parasites with up to 15 different species inhabiting the gut tract. So it's pretty common for gut parasites to find themselves in the presence of other species and for competition to happen in those habitats between those species. And it isn't just guts. Wherever parasites are trying to access the same resources, there is the potential for them to be in competition with one another. Sometimes it can be hard to wrap your mind around the fact that the gut is basically a forest. The gut is a coral reef. All the interactions that you'd expect among species in a forest or on a coral reef happen in the gut as well. Competition, both intraspecific and interspecific, plays out exactly the same way, except among towering trees or beautiful corals, it's among the microvilli and the twists and turns of the intestine. The ecological rules that apply to free-living species also apply to parasites, and competition is one of those realities that parasites have to face, just like free-living species do. I'll see you in the next video.